From Microbe TV, this is Immune, episode number 35, recorded on September 22nd, 2020. I'm Vincent Racaniello, and you're listening to the podcast about the body's defenders against disease. Joining me today from Durham, North Carolina, Steph Langle.
Hi, everyone. I'm back.
Welcome back.
It's been a couple months. Yeah, I appreciate you all, going forth. The podcasts were great. I'm sorry I couldn't be there. I had a baby in mid-July amidst a pandemic. And so I was able to keep a lot of things going, wrote a couple grants during maternity leave, which I don't promote doing that, but it just was a circumstantial thing. But the SciComm, I just couldn't keep up. So bravo to you all for keeping this going.
Also joining us from Ithaca, New York, Cindy Leifer.
Hi, how are y'all doing?
Okay. And from Madison, New Jersey, Brianne Barker.
Hi, it's great to see you all.
So, Steph, you were last on Immune what month? January-ish?
No, I did. I was on through the pandemic. So I did not.
That's
May,
right,
I
of
think.
course.
Yeah. So
May or June, right?
May or June.
Right.
It was the last two that I was not on. So.
All right. Yeah, that's right.
But I know, time flies.
You did a lot of SARS CoV 2 immunology.
The SARS stuff,
Yeah. Okay, now I remember. Well, we're glad to have you back. And
Thank you.
now we are complete. As someone wrote, what was that term? Immuno what?
So in an email?
Immuninati.
Immuninati. That was my favorite one. So
That's
far.
good.
I love it.
Dear Immuninati. Yeah, that was great. I really liked
We
that.
need like a triangle where
We're
we're illuminating everyone.
Yeah, the Illuminati.
something for the conspiracy theorists.
That's what it's based
Exactly.
on. Yeah.
Exactly.
All right.
I think also we should probably officially invite Brianne to be a part of the Immune team. I mean, more than 3 months running now.
Yeah.
Yeah,
Yeah.
well, thanks.
Can you keep doing two? Because
I was
we
going
would
to
love
say,
keep having you.
I think I can. I don't know if I can always keep doing TWIV twice a week. But
yeah.
I can't listen to TWIVs fast enough. You guys are doing great, pumping out.
Yeah, I
a lot of
understand. Two a week is hard, so you do whatever you can.
I gave the first of my chat with a virologist talks today.
You did?
Mhm.
Yes. I think I have gotten 10 requests so far.
Good for you.
Good, very good.
And
But you're an immunologist, aren't you?
I know, but they keep picking me. It's very strange.
Are you a viral immunologist?
There you go.
I
Now
think
you're
I am
connected.
a viral immunologist.
Now you're good.
Yes.
Good.
I did a seminar at the University of Kentucky yesterday. It was fun.
Huh.
Did your audio work? Because my audio also didn't work this morning.
And it did.
But I could not hear them. So
Yeah.
they had to ask me questions in chat that I could respond to.
That's too bad.
Oh, no.
Yeah, so I did a
sucks.
usual thing where I met with faculty all day. I had lunch with the students, all Zoom, and
All Zoom? Look
then
at this.
seminar. And then after the seminar, we had a cocktail hour, but and one of
you
the faculty, she
partake?
was home. She had her laptop in the kitchen and she was cooking. She said, "I'm sorry, I have to cook dinner while I talk to you." It was really funny. It was fun.
Yeah, it is. But it's going to be interesting moving forward with Zoom culture and how we're going to,
Yeah.
I mean, let's say there's a vaccine, it's efficacious at a certain percent. Do we go back to all these in-person things? It's really restrictive in terms of conferences for the cost of particularly smaller labs. And so if you're on Zoom,
Yeah.
you
Well,
remove that.
I think for smaller places that can't afford having visitors, they'll do it.
Yeah.
Because they've
Yep.
probably discovered it now. But I mean, I love visiting it. I would have loved yesterday to go to dinner, right? And
Right.
because you sit around and you chat, and that's great. No.
I have to say, though, there's one nice thing that I haven't been able to take as much advantage of as I would have liked, but my trainees and other students in the department is a lot of the big institutions are making their seminar series available for free.
Yeah.
And like the global immunotalks and the Inflamazoom. These are just, it's fantastic to see the biggest people that you might not be able to bring to smaller institutions, but you can hear them talk about their latest research.
Yeah,
so
I think
that's
that'll
been
stay.
really nice.
That'll stay for sure.
Yeah, I remember the seminars really fondly from being a graduate student, sort of the Harvard Wednesday seminar, and now it's on Zoom. And so I get to sit there every Wednesday afternoon at 4 and be part of the seminar, just like I did as a graduate student.
Yeah.
Yep.
I like, for us, we get lots of guests easily on Zoom because now people are used to Zoom. Here, I send you a link, right? So it's no brainer. It's just before it wasn't that typical for people to do that. And it was always, we used Skype and it was a little bit harder. So they had to have a Skype account, blah, blah, blah. So I think we're going to just keep having lots of guests. It's cool. I like it.
Cool.
Great.
I don't really miss traveling. I don't miss going to a restaurant. I don't miss all these things that some people seem to miss. I can wait. And if I have to wait a year and a half, 2 years, that's fine. I don't have a problem.
I think everybody has a different tolerance level
for
that
sure.
they can wait. Like some people are already way past their limit, and then other people are like,
everybody's
I can wait
different.
a few more months.
I agree.
I'm starting to get a little antsy. Once winter comes and we're all stuck inside, I don't know. We've gone out to dinner a couple of different times and eaten outside, and now they start to have those heater things so that they continue to eat outside, but it was kind of cold the other night. And I'm like, I don't know if I can do this anymore.
Hear that
in Ithaca, that's going to happen fast.
I hear that those are hard to find now, those outdoor heaters.
Doesn't surprise me.
Everybody's buying them.
Doesn't surprise me.
If you like what we do here on Immune, go over to microbe.tv/contribute. There are different ways you can help us financially there. Today we have a data paper and a review article that's related, which is pretty cool. So let's do the data paper first. This is a bioRxiv preprint, and it is called SARS CoV-2 infection severity is linked to superior humoral immunity against the spike. And this comes from University of Chicago, Washington University, Mount Sinai here in New York City, and Argonne National Laboratory. Let's see, these authors contributed equally. First three authors, Jenna Guthmiller, Olivia Stovishek, and Xiaolong Wang. And then the last author is Patrick Wilson. But here there are people I recognize like Florian Kramer, David Fremont, Yoshikawa Oka, and Patrick Wilson as well. Anybody else?
Yep, and I recognize Christopher Nelson. I think he just got a position, I think it's Stanford Immunology.
So this is an example of these multi-site collaborations, which are very common in SARS-CoV-2. Anyways,
and I was just reading, there was a great mini review by Patrick Wilson and Gabriel Victora on germinal cell development during influenza infection. So they, I think, have a lot collaboration going on, but then, looking at the germinal center in response to SARS-2 infections, highly relevant to what's going on here. So interesting.
No, it is still
there's
early.
a recent paper on that.
Yes.
In Cell.
So many papers.
So it's still early, right? It's only 8-ish, 9 months. So it's not a long-term anything, but we're slowly learning. So this is, as the title says, looking at the antibody response. And I thought I would just reiterate SARS-CoV-2 encodes 4 structural proteins, right? The spike, which everyone in the world knows about, I guess. The nucleocapsid protein, or N, which coats the RNA in the particle, right? The RNA is coated with a protein. And that's kind of unusual for a plus-stranded RNA virus. The coronaviruses, the nidovirales, they all have this retroviridae as well. But poliovirus, for example, which is sitting on the couch behind me, spilling out its RNA, that RNA is naked in the particle. So for the coronas, we think it's coated with protein because it's so long. All right, so 4 structural proteins: spike, nucleocapsid, matrix, and envelope protein. And then there are 16 non-structural proteins. They're called NSP1 to NSP16. And then there are accessory open reading frame proteins like ORF7 and ORF8. So, when you potentially all of these can give rise to antibodies. Right. So what they did here.
We would expect, though, that more antibodies would be generated to surface proteins, right?
Yeah, some are immunodominant. I think spike and N are immunodominant, and the others less. So N particularly, as you'll see, there's a lot of copies of N in the virus particle.
Yeah.
There are
and
much
if you think of the virion, so for those who are maybe joining us for the first time and haven't really gotten to know SARS-CoV-2.
There it is. Very nice.
There it is.
Now,
There
what
it is.
protein makes up the eyes, by the way?
That's a good question.
Is that the E protein?
I guess so.
Very good. What are you, a Sesame Street?
The
E is for eye.
protein, no?
I could be I or E, yeah.
Yeah.
Oh, right. Yeah. But the yellow, so that's the spike. So obviously it's accessible to antibodies more likely than the nucleocapsid, which is going to be inside the virion, which is coating the RNA, like Vincent said, probably to protect it because it's such a huge genome. But then the other proteins, there are antibodies against the M, but not against E. And that's pretty conserved, I found, because I studied a porcine coronavirus and we could never find E. Yeah. It's not immunogenic.
Interesting.
For whatever reason. So.
Yeah, they talk about this really nicely, just as Vincent said, about how there are different numbers of copies of all of these proteins.
Yes.
And that may be related to this. And they do the math of how many copies of N versus about how many copies of spike. So my question, I would wonder, Stephanie, is whether that protein that you couldn't find was there in many copies or not.
Yeah, I don't think it's as high as nucleocapsid, but I think it's maybe similar to M because it associates with the envelope and matrix. But I guess immunogenicity is in the way the protein presents itself is probably why.
So I guess one of the things that we could talk about is why are particular proteins, why do we generate antibodies to particular proteins more than others? So one could be the amount of total protein that's there, right? Mm-hmm. The second is the accessibility, because B cells express a membrane-bound version of the antibody on the surface, and they'll bind to whatever it is. And so if the antigen to which the antibody is going to bind is available, it'll bind to it more easily and then pull that into the cell, chop it up, and then get some help from T cells, right? So the other thing that's really important is that has to do with immunogenicity is that the peptides from that you have to have the antigen site available for the antibody to bind, but then you also have to have cognate associated peptides that are well presented by MHC to activate T cells. So when you take up that antigen and you chop it up into little bits and you put it into an MHC molecule to activate a T cell, that has to be efficient too. And so you have to have some sort of T cell that's activated in order to be able to provide the help. So it's a combination of all those things that will determine whether or not you generate high levels of antibodies. And how much of the virus replicates also. So how many total copies of the virus do you have? Right.
So one thing that sometimes people underestimate is that you can get antibodies to internal proteins.
Yep.
Usually that might have to be after the virus has come apart to make those proteins accessible. But I think sometimes people think that it's only the ones on the outside that are an option, and that's not the case.
Well, the non-structural proteins, which are not even in the particle, right? Those are made in infected cells, and those can be processed and displayed, presented as well. Mm-hmm. So for those of you looking at the video, I put up a picture of a generic coronavirus, so you can see the spike, the bilayer, the M protein, and the E glycoprotein, the N, and then the hemagglutinin esterase, a protein we talked about last Friday on TWIV. Which is not in SARS CoV-2, but it's in the seasonal coronaviruses. Do the animal, Steph, the animal coronas have the HE as well?
Yeah. Yep, they do. Mm-hmm. Yeah, some of the bovines do, not all the swines. It depends on the lineage. So there's alpha and beta coronaviruses, and then within each, they have lineages A through D. And so depending on where they fall, you can evolutionarily look at the way probably viruses mutate and if they lost that function or not. The other thing that's interesting in terms of developing antibodies against particular proteins is the kinetics of when it's produced in the cell. So copies matter and how much of the protein, but when in the process. So I think we'll talk about that the N protein is produced before the spike. And depending on your reactions of your B cells and all of its other cells it's working with could play a role.
So this is a study of patients. They had plasma from 35 hospitalized, acutely ill SARS-CoV-2 patients, and then 105 convalescent subjects. and they did ELISA assays for antibodies against spike and ORF7A, ORF8, NSP3, 9, 10, 15 proteins. Okay. Now, they make a statement that I found puzzling. Maybe you can explain it, or maybe you don't find puzzling. They say, notably, all subjects within the acutely infected cohort were hospitalized. Yes, but that's because you picked hospitalized patients, right? Because then they say, and in contrast, only 9% in the convalescent had ever been hospitalized. It seems to me that that's not a comparison, because you're getting samples from hospitalized patients. Am I missing something?
No, it's a weird way to word it, and I'm assuming the editors will have them clarify that.
That's true. This is a preprint.
This paper, because I'm assuming that's just you couldn't compare because it's not randomly selected. It's not evenly balanced. I think they divided into high, medium, and low responders, but you can't really. There's not going to be enough numbers in each of those groups to really look at hospitalized or non-hospitalized.
Yeah, I mean, I think it's important that they point this difference out, but the way they phrased it is that that's an interesting comparison that I'm not sure you can make.
No, because you're picking hospitalized patients, so that by definition they're hospitalized, and 100% of them are, right? Because if you're only anyway, 89% of the acutely infected subject, 98% of convalescent had antibodies against one or more antigens, and nearly all of them had spike and N protein antibodies. And the convalescent subjects also had a response against the receptor binding domain of the spike and the RNA binding domain of the N protein. And they say maybe these have the immunodominant epitopes. More acutely infected subjects had antibodies against ORF7A, 8, and NSP, these non-structural proteins that are inside the cells. And they say maybe antibodies against these are short-lived or induced by more severe infection, because they don't find them as frequently in the convalescent subjects. And then, as Steph mentioned, the anti-N antibody response preceded the response against spike and was consistently higher across all the time points, peaking between the 2nd and 3rd week after symptom onset, and then declining between the 3rd and 4th weeks. They also
this seems pretty standard for most infections, the way we learn it in our basic immunology classes. So it's not out of the ordinary.
Yeah. They found a strong positive correlation between anti-N and anti-G titers in both sets of patients, and they conclude that subjects who generally mount a robust antibody response tend to mount a robust response against both antigens. And the antibodies against the spike and persist 2+ months after symptom onset. And that's also sort of consistent with other studies, I think, that we've talked about. Although beyond that, who knows? Probably starts to go down. So
I would imagine, and based on the review we're going to talk about, a lot of other coronaviruses do go down. But also you're thinking about the time course of this pandemic. We're only 9 months out, so we don't really get to know that information quite yet. I get a lot of questions about, immunity, and is the vaccine going to provide durable immunity? And so these things just take time.
Right. And the other thing to point out is that if you look at an immune response in that basic immunology class, we see that it goes down from peak. The antibody number, the T cell number doesn't stay at the highest level that it achieves. It has to come back down, and that's a really good thing, so that you have more abilities to make other immune responses later.
Right. Yes. I think we do have this obsession with, we want these high titers. We need high titers. They need to be long lasting. They need to be durable. But if your blood and your germinal centers in your lymph nodes are filled with B cells producing this one clone or this one specific antibody, and you get infected with influenza or another virus, there's not going to be the opportunity to provide antibodies for that virus. And so I think that's the body's way of making sure you have the capacity to provide protection for a variety.
Yeah. So we have to think not just about, is there a decline, but is there a decline that's more than normal or different than normal?
Yeah.
So Steph, over on TWIV, we've had an ongoing discussion about immunity, because you could be immune to infection or immune to disease, but not infection, right?
Right.
And when people
Right.
say, am I going to be immune? Well, I say, what do you mean? Do you want to be immune to infection? That's pretty hard with the coronaviruses. You know,
It is, and I think
With
that's why
a lot of with pretty much anything, right?
Yeah,
I mean,
that's
it's
true.
difficult to generate enough mucosal antibody to protect you from a pathogen from ever
Absolutely.
invading. So
Yeah.
sterilizing immunity seems
It's very hard.
You know,
both polio
very, very
vaccines allow infection of the gut. You know,
Yeah.
so it's hard. By the way, now the sun is coming down in the sky, and I've got my
I
headlights.
see. You do. You have your hairlight.
There you go.
You are illuminated.
Yeah, I'm not an immunity, but I'm an Illuminati.
And the other interesting thing too, you think about immunity and persistent duration of antibodies. That's really looking at serum, but what we're trying to do is stop transmission at the mucosal surface, which is any parts of you that produce mucus, and that's where the virus is going to bind its receptors. So we really need antibodies there. But we don't measure those because it's hard to get.
It's hard enough to stick a swab up somebody's nose, to do nasal washes to get all the antibodies, and then have enough there to be able to detect the titers. Pretty limited.
And I suppose
you can usually measure fecal IgA, but I don't know how that usually.
These very sick patients probably don't want to be doing nasal washes, right? You have enough trouble. They may be intubated anyway, right? So So from this part of this study, they conclude that the antibody is largely driven against the spike in the N, and the anti-N is first. And they say, well, maybe differences in protein production, N covering the entire genome, and a single particle only has 26 trimers of the spike protein. So that could be part of it.
And how many N? Was it something like 1,000 per viral particle?
Yeah, I think it was 1,000 near the end.
Okay, so then they said, can we compare responders? Can we divide these individuals into responders, cluster them into groups based on antibody titers, right, against these various proteins? So from the acutely infected patients, they had 3 clusters: high, mid, and low responders. And they say the high responder were further from the onset at the time of sampling, and they ultimately were hospitalized for a longer duration than those in the mid and low responder group. So that's kind of what's in the title, the idea that high antibody, high or more ill patient, I guess. And in this group, the acute, there's no difference in age or sex between the 3 responder groups. So they say it looks like these high responders had more likely to have severe infections.
So this is similar to some of the other studies that we've talked about and some published things. But do you guys believe that this study has enough power to make these determinations?
No, I don't think so, right?
It's 35 patients, right? And they've divided them into 3 groups.
Yeah. I think they can't. I think it's a
I don't
suggestion.
know. I think it's a little suspect to make definitive conclusions.
Yeah, I have lots of questions about sort of some mechanistic stuff, but you're right.
I think
there's also some questions about power.
They're measured. They say suggest a lot, right? So I don't think they can conclude that. But this is very typical. You often see these low number studies, right?
Yeah. So do you think you want a lot of antibody or a little antibody, right? Because this is the conundrum, right? Because one would, intuitively think more antibodies are better.
Sure.
And certainly if we immunize with a vaccine, we would think that more antibodies would be better because it would protect you early on during infection.
Yeah.
But it seems like paper after paper that are coming out, the more robust the immune response, the worse off you are.
Yeah, but I'm not sure that it's causative, right? I mean,
exactly.
I think,
that's been the thing I worried about.
I
was wondering about.
In the accompanying review, they say that one of the ideas, which I like, is that, the people who are really ill, the virus is reproducing to higher titers. You have more antigens, and maybe that's what's driving higher responses.
So I've kind of wondered, is what's going on here that having an increased antibody response is leading to worse disease, or are you having more virus or virus longer, which is leading to both an increased antibody response and more severe disease.
Yeah, I think that right, two different questions. And to go back and just because the papers come out so quickly, maybe I missed it. In a general population, we cannot prescribe a viral titer to a risk of getting severe disease. Correct.
Yeah,
but I know that there was a paper that at least for if we just take the subset of hospitalized patients within that cohort, viral titer did matter then.
Well, well, there are two things. First, the virus moves down into the lungs in those serious patients, and then it persists longer than in the moderately infected patients. Right? There's a timeline where titers drop really 7 days after symptom onset, and then that's it. Whereas these very sick patients tend to shed longer, but not forever. Right? They don't shed. So Daniel Griffin said last time on TWIV, he has a woman who's been in hospital for 2 months, and she's been negative for a long time. She just has subsequent issues, like her lungs are destroyed and she can't get off oxygen, right?
Right. So it's interesting, and I know Cindy and Bran will appreciate this being loving innate immunity, but I do. So if you have, let's say, across these patients, the viral titers don't differ that much in terms of peak or length. Likely, then it's an immune-mediated difference between the people who have higher or lower. So if you have more damage to cells, you have more of these danger signals being produced from the burst open infected cells, then you probably have more robust germinal center reactions in the lymph nodes. So maybe it's not so much there's so much more virus, but that the body has responded so much more that those cues are making the germinal center say, hey, we need to make these B cells produce more antibody, potentially.
I think that's a great possibility.
Yeah.
Agreed.
All right. So continuing with the stratification, high responders had a larger response to N relative to mid. I also, the high and mid responders more likely to make antibodies against Orphate and NSPs. So they say acutely infected subjects, these are the hospitalized ones, mounted larger. And I find this weird that they call it a larger antibody response. I don't think that's the word that you usually use, right?
Be higher or lower.
Yeah. Against N protein, we're more likely to mount a response against other antigens. Okay. Then we have the convalescent cohort. They also could put them in 3 clusters based on the spike and N antibodies. And, but of course they were convalescent. Only a few of those were in hospital actually. So then they say, can we make a correlation between infection severity and antibody responses within this convalescent cohort? There, they have to use self-reported symptoms, which is always problematic, right? Because,
Yeah.
You know, what makes me feel badly is different from other people, and whether they're hospitalized, which is a small number. Over half of the subjects within the high responder group of this convalescent group had a severe infection. And subjects within this high responder were typically older and male. So those are the 2 risk factors that we know of for more severe disease, which is interesting. And you remember that hospitalized subjects, there was no stratification by sex. It's quite interesting. And within the high responder group, within this convalescent group, they had higher titers not only against N, but spike in Orphate as well, relative to mid and low responders. So they're more likely to seroconvert against one of the other proteins as well. So that they conclude that the two cohorts, the convalescent and the acutely infected, those with more severe infection within both groups mount here again larger antibody response at both the acute and convalescent time points.
Yeah. Do you wonder really what was it necessary to define the acute and the convalescent? Because it does seem within each of those, there were varying timelines and the result was the same between the two. So we really didn't learn much about those 2 groups. Yeah,
it's just the magnitude, right? The ones in hospital, all of them had this higher grouping of the antibody responses. Yeah, I don't think we learned all that much, right? Then they looked at memory B cells by taking peripheral blood mononuclear cells and doing Ellis spots. And so they say these memory B cells largely were against the spike, very few targeted N or ORF8. And this is from the convalescent subjects.
What do you guys think about sampling those memory B cells from peripheral blood?
Right. I mean, probably the ones that are resident within the lung or the nasal passage is going to be probably more reflective of what's going to be protective.
Yeah, it was sort of, I was really excited, I think, after some other paper that we read in the past for one of these podcasts to look at how they had defined memory B cells.
Right.
And here, memory B cells basically meant B cells that could respond to antigen in culture.
Yes.
Yeah. So that's not so robust. I think the paper where we talked about the memory B cells before, wasn't it with the measles virus eliminating the memory?
Michael Minna, that was his first appearance.
Minna, yep. Mm-hmm. Yep.
So Cindy, would you rather?
They did a really nice job of defining what a memory B cell was. And clearly we know some of them circulate, but the vast majority of memory B cells are going to park themselves in tissues or back in the bone marrow. Right.
Yeah. So would you rather have bone marrow?
Well, I don't think that that's really a sampling opportunity for humans all that frequently.
Yeah.
Right.
But it raises the question of, when you sample in a mouse model, you can find these things more easily because you can harvest those tissues, the spleen or the lung or the bone marrow. And in human, we kind of have to deal with what we have available to us, which isn't always the most ideal.
Yeah.
Yeah. So I think you raise a really important issue about, are they really looking at a memory B cell?
Yeah. I would say they're looking at, B cells that are functional.
Yeah.
B cells that are antibody producing cells. And I'll leave what we call a memory B cell to others who spend more time thinking about those.
Well, right. So in the blood, for those who may not have heard us talk about B cells before, after you have an infection and you have a piece of that virus or bacteria being presented to the B cell to produce antibody, those B cells they have a fate. They have a differentiation fate. They could either be these really short term, they're going to start producing antibodies quickly. Those antibodies are not going to be super specific, but they could turn into long-term plasma cells. This is the cell type. So plasma cells is one fate. The other fate is these memory B cells. They are going to clonally proliferate. So multiple of them are going to start to traffic to likely the site of infection and retain there and become resident memory B cells. And so what Brian's talking about, they're going to circulate, but the way that they looked at them might not be a robust way to say, oh, these are memory, or these are just B cells that produce antibody. Yeah. And it's easy, of course, for us, for these type of papers to have little, we wish they did this and this. We understand, of course, this is sometimes the only thing they could do at the time, but.
Yeah, I will also point out that I have this a little bit on the brain since I'm listening to a PhD defense in 2 days on resident memory B cells.
Look at that. So, now, where are they resident to? Is she or he focused on a specific organ?
Lung.
Oh, look at
it's my sister.
Oh, how exciting.
Yes. That's great. Where is she? She's going to is she at NYU?
She's at BU.
BU. Okay.
Yeah.
Nice. Good.
So I have been thinking a lot about resident memory B cells.
Wow. What a perfect time for her to be. You said she's defending?
Yeah.
Oh, wow. Okay, well, good luck. Yes.
So the authors say this memory B cell response is largely directed against spike. Then they look at this amino acid variant in the spike, which
actually, do you guys want to go back to that because we know there's more. So there's more antibody to N protein.
Yes,
but the memory, quote unquote, they're calling memory is maybe to spike.
Yeah, yeah, right.
And so I guess that relates to this concept of like immunodominance and what protein is selected for which B cell fate. And that's going to be really important for vaccine design.
So it makes sense if these were really truly memory B cells that you would want to keep the ones around that are going to be more protective
Mm.
And
Right, 'cause-
so we would think that you would select more for those anti-spike protein antibodies because they would be more likely to block or target the virus for elimination.
Right. Which we don't get to really learn about the effector function, the neutralizing function.
No.
Sorry, Vincent.
No, it's okay. No worries.
But you're right.
I want to skip over this. They have a section on the D614G spike variant, which I find very uninteresting. And so,
Is it your favorite topic?
yeah, I mean, I just don't think this has been shown to.
I'm not sure I totally got what they were saying. I think they were saying that the convalescent cohort had more likely been infected by the original one that had more cross reactivity or something like that. I didn't quite get exactly what they were saying. But
and with those numbers, I don't know if it.
Yeah, I think the only take-home message I had was that the antibodies didn't seem to care between the two variants.
Yeah.
But, in terms of the relevance of, so for those who are not familiar, I mean, this is a mutation that happened in the spike protein. We're not going to comment on whether it makes it more or less transmissible because these are things that take experimentation to find out. But you can follow the antibody response against this region of the spike and understand, does that affect neutralization? Does it affect, in culture?
Yeah.
In vitro?
Well, antibodies, this doesn't affect neutralization with antibodies. They say this area with the change is not immunodominant, doesn't affect local epitope antigenicity. Whether antibodies targeting the receptor binding domain cross-reacted with, this is a separate question, RBD proteins of other coronaviruses, including SARS-1 and MERS coronavirus. So they found, I think this is what you were referring to, Cindy, positive correlation between antibody titers against SARS-CoV-2 receptor binding domain and SARS-CoV-1, but not MERS receptor binding domain. Maybe not surprising because SARS-1 and 2 both bind the same receptor and MERS a different one.
Binds a different receptor, right?
Yeah.
Yep.
Right.
All right, so those are the data. Sorry, go ahead.
Oh, no, it's fine. We'll probably talk about it in the review paper, but the concept of cross-reactivity and why it matters and why we're so obsessed with it right now.
Yeah, they do talk a bit about it in the review paper. We'll get into it there. But basically, they say subjects with more severe disease likely have increased viral titers, free antigen in the lung, leading to increased titers. And they say epitope spreading may be a factor. What is epitope spreading? How would you define that?
I mean, I think it's the ability to have an increased antibody response against more than just the spike and nucleocapsid. So spreading amongst these non-canonical proteins, the ORFs.
Yeah. So I would say it's a response that starts against perhaps one antigen and then broadens to be against other antigens. It spreads to those other antigens.
Okay. So if you have more viral spread within the host, more replication, then that would lead to that more increased response.
More buildup of the other proteins.
Okay. Yeah, especially if you have a lot of cell death that might expose some of the antigens that hadn't previously been exposed.
Makes sense.
It also, I think, could happen if you start to generate antibodies early on during the infection to, let's say, spike, and that allows you to internalize more of the virus and present more of the antigen. And so that might allow you to start to recognize other epitopes as well.
Okay.
This is a really important concept in disease, like certain autoimmune diseases, where, like in lupus, where you start out with an auto antibody against one antigen, but as the inflammation persists, the chronic inflammation, and you keep presenting and taking up more antigens and presenting them and feed forward this loop, you start getting another wave of antibodies to a different autoantigen and another wave of antibodies to a different autoantigen until you have many autoantigens involved in the recognition.
Got it.
Yeah, which I think, and they did maybe discuss this, but when during a viral infection, so if you have persistent inflammation particularly for people who cannot clear or who have immunopathology that's causing severe cell death, you're going to find more antibodies against muscle or cardiolipin.
You can break tolerance.
Right.
Yep.
Okay. So Go
For this
ahead.
paper, I mean, so that kind of overarching, why does this matter for immunity and SARS-CoV-2? I think when we're thinking about correlates of protection, this would actually, be the opposite, right? Because you would think, well, if they have higher amounts of antibodies, they should be better protected.
Right. Yeah.
But that's not necessarily the case because one, these are severely ill patients. So we're not really looking at asymptomatic individuals.
Right.
But trying to define, if you pull someone's blood and you do a quick antibody test, what does that mean? And that's what these studies are really trying to get at. What can we predict from this?
Yeah, in fact, they asked the question whether subjects with more severe disease are more likely to be protected from reinfection, right? But I think it's not likely because their titers are going to wane eventually as well, just because they're higher. It may take a little bit longer. But I think protected from reinfection isn't right. It's not the right way to look at it because all the endemic coronaviruses reinfect frequently. We did a paper on TWIV last week. Every 6 months in this one Amsterdam cohort that they studied, reinfection every 6 months. Antibody titers go down, get reinfection, they go up again, they go down every 6 months, mostly in the absence of disease. So
that's what I was going to say. Is there symptomatic disease?
Yeah, most of these, there is disease caused by these viruses, but it seems to be in certain individuals, frail individuals, as they said, they called them. And so I think that's the rule for these human coronaviruses, reinfection, less disease or no disease. So I don't think we should talk about protection from reinfection. I don't think that's going to happen. But that's kind of scary for a vaccine because if a vaccine doesn't prevent reinfection, if it prevents disease, that's great. But if you're shedding and you can transmit, that has huge implications, right? It means everybody has to be vaccinated.
Yes, everyone would need to be vaccinated. What it would do is provide protection against the most vulnerable, because they're the ones getting the disease. So that would be protection against disease in those individuals. But I think, especially with kids, which we're learning shed high amounts of virus, can be these silent spreaders. And I don't know if we're even testing kids right now in clinical trials. So I agree with you. And also in terms of reinfection of this cohort, their lungs are probably so damaged that it wouldn't be a cohort that you would want to look at reinfection because there's going to be a whole host of other problems with them that would make them more susceptible probably.
Yeah. We should be careful though. Just because we see a particular type of immune response during natural infection doesn't mean that that's going to be what we'll see with a vaccine. The vaccine could lead to differences. Maybe there's some protein that's modifying immune responses in the virus that's not present in the vaccine. And so, we don't have to worry about that modification. Or you could think of a million different ways those could be different.
Yeah, I think that's correct. I hope that's true, but I just have never seen a vaccine that did better than natural infection. It's just it's not the wild type virus. So you can come close, but so I'm hoping you're right. I hope that the virus is doing something that a spike subunit alone is not going to do. And maybe you'll get durable immunity, but we'll see.
Sure.
Right? We'll see.
And I know that's frustrating for a lot of people, when they ask about this question of immunity, because a lot of people are struggling. They want to understand, when is this, over? I don't know if that's the right way to be thinking of it. It's really going to be this marathon of, you're going to see every season, probably like the other cold and respiratory viruses, this is going to recirculate. But if we've already implemented kind of this culture, maybe of mask wearing potentially, and then the vaccine, if it provided like 50% efficacy plus these other measures, but it's really difficult to know. I don't know. The vaccine is not necessarily going to be the end of worrying about this problem. And I know that's really hard to hear.
Well, Tony Fauci said we'll be normal maybe at the end of 2021. But the virus is never going to go away. It's going to be with us forever.
Yeah. Normal would be hard to define based on
the new normal.
Yes.
Yeah, it's funny you say mask wearing. I complain on Twitter. So many people wear their masks,
Oh,
down
driving crazy.
and then when they approach you on the street, they whip it up, and
they're all wearing them here under their chin, and they just pull it up and pull it down.
I was getting gas this morning. Across the street, there's a daycare center, and people are bringing their kids in wearing masks. They're getting a fever check. And then the parents go back to the car and they rip their mask off and throw it in the front seat, total not doffing the right way. But
viral particles everywhere.
Yeah. I mean, these are potentially contaminated masks. You have to take them off a certain way. You should fold them carefully, right? And, oh, well, at least they're wearing them, right?
Right.
So the second is a review article. This is published in Nature Communications, Systematic Review of Antibody-Mediated Immunity to Coronaviruses, Kinetics, Correlates of Protection, and Association with Severity. The things we were just talking about really in that research paper. And this
is a tour de force.
It's a big paper. So we cannot cover everything, but we'll make some highlights. But it's
I haven't seen a lot of papers done like this. It's sort of a meta-analysis, and sort of a review
it's mainly,
of a lot of different studies.
It's mainly the other coronaviruses, other than SARS-CoV-2. There's a little bit of data from SARS-CoV-2, but this is from groups at the University of Florida, Armed Forces Research Institute in Bangkok, National Institutes of Health, UCSF, Johns Hopkins, University of Cambridge, and the Pasteur Institute. And first author, Ankana Huang. The last author is Derek Cummings.
There are actually 5 co-first authors.
Yeah.
Really?
So Ankana Huang, Bernardo Garcia Carreras, Matt Hitchings, Bingyi Yang, and Leah Katzelnik contributed equally.
Do we have co-PIs too?
No.
Good. Okay. Now, the thing is, these journals sometimes hide that at the end, so I can't see it when some of the journals put it right at the bottom, but others don't. All right, so this is a literature review, basically, and try and learn from what we know about other coronaviruses and extrapolate to SARS-CoV-2. So what they did here is they focused on the human coronaviruses, the endemic human coronaviruses 229E, HKU1, OC43 and NL63, SARS-CoV and MERS-CoV, and of course, a little bit of SARS-CoV-2. They found 2,452 abstracts and were all read by 2 reviewers, and they identified 491 papers for full review. I guess all these authors read all these papers and summarized them. And so they're focusing on antibody responses in this review. So they look at papers that have used ELISAs, Western blots, et cetera, neutralization assays to pick up antibodies. And then it's divided into sections. So the first is the kinetics of the response and the clinical severity associated with it. And very nice figures that you can look at.
Beautiful figures.
This should be open access, I think. You can look at all of these. So let's see. Let me highlight some things, and you guys are going to see things I don't pick up, so just chime in. So the endemic coronaviruses, the 4 common colds, well, actually all of them. Antibody responses rarely seen during the acute phase, the first 7 days. Very rarely see antibody responses. Mostly you start the 2nd or 3rd week after symptom onset. And they have a very nice figure that actually compares MERS, SARS, and SARS-CoV-2, IgM and IgG neutralizing responses, and they stratify that with severity of infection, not reported mild or severe. And so you can see the kinetics and I think they say that SARS-CoV-2 kinetics are more rapid onset than the other 2 viruses, right? And you can see they're looking at days from symptom onset, and you can see there's a peak of antibody titers at a certain time, and then they all decline. And by 30 days, zip, nothing left, right?
Yep.
They're also a nice graph.
My Roomba decided to say something to me in the corner over here.
That's okay.
You heard that. Sorry about that.
She's absorbing passively information. Right?
Yeah, I guess she's talking over there.
But she can't hear us, though.
Right.
But they also have a nice.
So I feel like this, this meta-analysis, the way it's done, is again reconfirming what we know about how immune responses work. It takes roughly about 7 days ish to generate a robust antibody response that peaks about two weeks or so and then wanes. Then memory B cells are made and they persist. So it's like, okay, great. Yep. Exactly what we expected to see. Yeah. So that's good. I think what was also nice is that you talked about that onset of antibody. And I think it fits a little bit with, the replication and the transmission that we think happens. Yeah. With the MERS, I think the transmission started after you already had symptoms. And so, maybe the antibody response took a little longer because the virus took a little longer, but it was easier to control, right? But the ones that the antibody response is happening early, probably because the viral replication is happening really early, you're probably transmitting earlier. And that's the problem that we're having, right? Because people asymptomatically or pre-symptomatically are shedding and transmitting.
Yeah. And it does, though, if we're looking at figure 2, which I could even, for fun, do you want me to share my screen?
Sure, you can.
Always asking that in Zoom meetings, can you see my screen? So what's interesting, we talked about in the other paper, they were only really looking at, so there's high, mid, and low responders, but they weren't stratified based on severe, mild, or asymptomatic. That was not information that we got to capture. And so from that paper, we would say that people with more severe disease were more likely to have higher antibody responses. But what we see for SARS-CoV-2, when we include people that are mild, or I would assume, actually, I don't know about asymptomatic people in this group. That's actually hard to know. But people who had more mild disease had higher amounts of antibodies, correct? That's what we would assume from this graph. So that would then suggest that if you're able to produce antibodies more quickly and at higher levels, you will be more protected than those who can't do that. They get more severe disease and end up in the hospital. And so that speaks to this, what's going to protect us, correlation of protection. And it seems to be, at least for SARS-CoV-2, although it's opposite for MERS, or we just don't have info.
I think MERS was just so severe and such a high fatality rate that we didn't have very many mild cases. So I don't know how we can really compare it.
That's a good point. And that is that characteristic, having so many mild infections, is very unique to SARS-CoV-2. And it provides us many problems because people are not going to they're not sick enough to go to the hospital and contain it. People are out spreading the virus. So, like Cindy said, this is very characteristic of what we would expect to see in terms of kinetics. But then people who have more mild disease had more antibodies.
Yeah. Well, if you look at the bottom on that figure, they've got 2 little dots with bars that I think are supposed to be the means for mild and severe. Yeah. And it looks like the sort of severe means might be a little higher, but I'm not sure any of that's significant.
Yeah. Oh, I see what you're saying.
Yeah. Is it higher, or is that just shifted to the right because it takes longer for the antibody response to develop?
Oh, that's what it is.
Okay.
But that is interesting.
Yeah.
They actually,
I don't know if that's statistically significant.
I don't think that's significant.
Though. I think you'd need more people.
Yeah, there is about a half a page discussion of this link between severity and antibody response. And they say the available information is uneven across viruses.
Right. Well, and the other thing about this review is they actually only included SARS-CoV-2 papers from March 22nd. That was their cutoff. So it's only 3 months. And actually since March, we've had a lot of papers, tons of papers. So
I agree.
That would be, but I know there's always a lag in publishing and you can't include everything to date.
Yeah, they make a nice comment at the end about how they tried to be really transparent with what they were doing so others could add on to it with papers that have come since March.
Yeah.
They have some data which suggests that even with the endemic common cold coronas, there's some suggestion that neutralizing antibodies are higher in severe cases. So there are some severe cases. They're not a lot. And so they say perhaps this is suggesting that even with those, severity relates to antibody response. Then they talk about correlates of protection, which we've mentioned, right? And they say, this is important, identification of a correlate of protection, like what kind of antibody response will protect you, requires characterization of immune responses before a known exposure or a period of risk in which infection or illness outcomes are characterized. And they found this level of detail was only present in human challenge experiments with the human coronavirus, the common cold, right? Because they're the only ones you can do that with because they don't kill you.
Right. Yeah.
They found 18 studies where they did these challenges, and you ask, so does having antibody protect you against infection? And they're only looking at antibody, right?
Right.
Mm-hmm.
Which doesn't answer whether T-cell immunity would protect you or not. But they mentioned, for example, that volunteers given higher doses were more likely to experience cold than those given lower doses, which makes sense because the lower dose never initiates an infection, probably. One study, lower proportions of individuals with high neutralizing antibodies experienced significant cold upon challenge than individuals with low titer, which, again, it's getting at the question, are antibodies the correlates of protection? And even some studies with antibodies, IgA and G in nasal wash, right? IgG and serum.
IgA was highly correlative to, associating with protection. Which makes sense if it's it's produced in gobs in the mucus, and that's where you have binding in the nasal cavity.
Yeah, they also have a study of antibodies among medical students, and they say pre-existing antibodies, and from these they either isolated virus or looked at antibodies. Pre-existing antibodies, neutralizing antibodies, were inversely associated with increases in antibodies after reinfection. But that means, in other words, they protect you.
Yep.
So basically, these challenge experiments, you can ask whether antibodies are protective, and in some cases they do, not in all cases, which just illustrates the difficulties in these kinds of infections because humans are all different. They're not inbred mice.
That's right.
And, as with animal challenge experiments, I mean, these are controlled where you know the dose.
That's right.
You're giving the dose in one route. Like, I don't know what they did, but let's say the nose. Whereas in real life, we're getting the dose could vary in different orifices, your mouth or your nose, your eyes even. So
totally.
I think that's what makes it challenging, when the question of does viral dose matter? I think, well, of course, when I gave pigs more of PEDV, they experienced more death, but in the natural ecology of the virus infection, it'd be very different.
Yeah. As they say, these doses may differ from the amounts of virus people are exposed to, for sure.
Yeah.
I mean, almost everyone gets a different amount, really. So
that's something that people will ask me. They say these masks are not 100% effective, right? Because the whole argument is that our masks protect others, not us, right? Because if we're spewing out virus, it's going to catch the vast majority of it. But then the question becomes, do they partially protect us? Because if it reduces the load that you might be exposed to, like say you're standing next to somebody who's shedding and you get some particles in and a few viruses get in you, would that be a good thing? Would you be able to mount a response? Is it enough? Or, is one enough to induce an infection? Or how many do you need to induce an infection? And what level of response do you need to then be protected? So is a little bit a good thing as you tickle your immune response, you get some antibodies and that will protect you. So a low dose exposure is a good thing. So are the masks protecting us? Are they not? So this is like this argument I get all the time in question, and I'm not 100% sure how to answer it because I don't know if we have enough information.
Well, we certainly don't know what the threshold is for. I mean, to get an immune response, you need to have some reproduction, right? If you get 10 particles in your nose and they don't reproduce, you're not going to make an antibody response. But
it depends on how much you get, right?
10 particles,
right? Oh, well, yeah.
For vaccines, you get a bolus. You get a lot of antigen.
You get a lot of it.
So inactivated vaccines, you get a lot of antigen. So,
but that doesn't require replication.
No, but it's a lot.
But if it's a natural exposure, you need some replication.
Yeah, so I think you need replication. In which case, if the mask is protecting you, giving you a sub-infective dose, then you're not going to be immunized by it. So I don't think that makes any sense. That idea that you get a little virus coming through and it's immunizing. No, I don't think that works at all. Well, we could do a challenge with common cold coronas. We could have people wear masks and spray virus at them and then see if they develop an antibody response. You could do that
Are
experiment.
you volunteering?
Yeah.
Sure. But someone has to pay for the experiment, right?
Right. Mm-hmm.
Then they do a literature search of cross-reactivity and antigenic diversity. And I'm not sure what the bottom line here is, that there is some cross-reactivity, especially among the endemic common cold coronaviruses. And sometimes inoculation with one will protect from challenge with another of these common cold coronaviruses. But overall, if you're living on Earth, you're going to get infected with one of these four. Some may be less prevalent than others, but in general, we see a lot of infection with three of the four at least. I think one of them is more rare than the others.
I mean, it is interesting in the concept. We think about it a lot in influenza, that, your first infection, I guess this is true for all infections, but they are going to affect every subsequent infection in terms of your antibody response. It's like the butterfly effect. You can never really know what could have happened. But the four coronaviruses, I mean, they're split 2 and 2 between two different A and B of beta or alpha coronaviruses, and they don't necessarily protect against each other. So the cross protection seems to be intra, not inter. So within each family. And SARS CoV-2 is in the same family as what that OC43. I'm looking at my notes, and the other one, I forget the name. One of the four.
Yeah, two of them are the same, and two of them are different.
Yes. And so there is the question of, do low levels, because we know they wane, of these common colds, do they impact, reaction within the germinal center that helps make the B cells produce antibodies to SARS CoV-2?
Yeah.
And we know T cells matter from what you all talked about last time.
I think there are a lot of arguments about this where people are trying to assess, do people have pre-existing immunity because of past infections with a cross-reactive virus, and how might that influence what's going on with what we're seeing in the pandemic?
Right.
And my answer is sort of like what Stephanie just said about the butterfly effect. Basically, if you live on Earth, as Vincent said, you've been infected with one of these. And so I'm not sure how we could actually look to see what happens if you absolutely never had been infected with one. What we're seeing is in mostly people who have been pre-infected.
I think you could potentially look at it in infants and know what their mother's antibodies, what they are too. And then you'd have to follow these babies very closely. And we know that young children are rarely, going to show disease, but it could be a population that you could try to identify cross-reactivity to SARS-CoV-2.
Well, the fact that SARS-CoV-2 is ripping through humanity tells me that there's no protection from infection anyway. Now, some people argue that maybe disease severity is somewhat related to your history, but as Brianne said, everybody's been infected, and I don't think there's any correlation with previous exposure to these common cold coronas and what happens now. I really don't. And remember, a lot of these studies are just looking at binding antibodies. They're not looking at neutralizing. So there's
right. And they mentioned that a lot of them, when they report the ELISA data, they don't necessarily report their cutoffs. And that's a problem when we talk about waning antibodies. The specificity of your assay, it's the variability becomes very great with the lower levels of whatever you're trying to measure. And so it would be very challenging without a very specific assay to know. It's a lot of false positives. Or could be.
So that comes to this idea that when you do a meta-analysis like this, the benefit of it is that you're looking at many, many different studies. And if you see similar trends, you can be more confident that something's true. But the deficiency of it is if you don't see those trends, it doesn't mean it's not there, because every experimental setup and every study has different numbers of patients with different backgrounds, and they're assaying different things. And as you're saying, Steph, with different sensitivities and cutoffs. And so it's really hard to do these kinds of meta-analyses to compare across studies.
Yep. They also show, for example, that these human coronaviruses can induce antibodies that bind some of the emerging ones, SARS, MERS, SARS-CoV-2. So, for example, SARS patients had antibody rise to the common cold coronaviruses. Animal handlers in Guangzhou, where SARS emerged, had antibodies to MERS. Of course, MERS hadn't even emerged. So they're just cross-reactive antibodies, right, of some class.
Right.
And their significance, we don't know. We have no idea
Right.
because these are just binding antibodies. So that's...
And do you know, I don't recall whether they were talking about when they sample those, because what do you think, Steph and Brianne, about the earlier on in the infection that you sample, you might have more broadly reactive antibodies, but then as the infection develops, you're going to start to develop that higher specificity, and you're going to have somatic hypermutation and so forth. And so you're going to have more specific antibodies that are less likely to cross-react in those individuals who either have been infected for a while or who have recovered.
Yeah, exactly. I mean, you usually see that sort of breadth of binding early and with more of an IgM type of response. And they really don't mention the details of some of that timing here, or whether they're talking about IgG or IgM. And so that could certainly be an issue.
Yeah, because it's going to take time for somatic hypermutation to happen in these interactions. And it's that iterative going back at the B cell consistently, being educated in the germinal center and the lymph node, and that makes them more have higher affinity. So you're right, Brianne. Even if it is IgG, if it's earlier, I mean, there are plasmablasts, these plasma cells that produce IgG, and it could be those.
Right.
So,
I want to move to the last two topics because I have to leave by 5 today.
Sure. One of these topics I'm excited to talk about, because this would be this question of immunopathology or vaccine-induced. So immunopathogenesis, can antibody cause disease? Antibody-dependent enhancement, the ability that an antibody bound to a particle allows it to infect a different cell, so you have a different outcome. And this is what happens with dengue, of course, with 4 serotypes. And you're familiar with this, Steph, because this happens with cat coronaviruses, right? Mm-hmm. It does. I mean, it was first found when they gave kittens passive immunity from their mothers, or I don't remember if it was like intravenous of just serum. I can't remember. But anyways, the kittens, when they became infected, they had higher rates of infection than the kittens that did not get passive immunity or antibody. And basically, it's specific for this feline infectious peritonitis virus because the receptor that the virus binds is on a macrophage. And it's those macrophages that also have this other receptor that kind of brings in the antibody close to it. And so when you have these complexes associated with the macrophage, then you have this enhanced disease and ability for the virus to infect more cells because they're closer to the cells that they infect. My question, and I'd be curious of your thoughts, there's a lot of people are scared that this could happen with this virus. Yep. In my reading, there's not really much evidence to be scared. It's just the fact that it could happen and there's going to be millions of people vaccinated.
I think that's correct. A rare event is going to be
really
amplified.
No in vivo evidence.
You're right.
That this is going to happen.
I mean, I was wondering what you thought of this infant study where, the highest lower tract disease in infants 6 to 23 months of age, whereas the upper tract burden was close and older. And they say, well, maybe the maternal antibodies were causing lower tract disease, right?
Right. So I took a look at that and the numbers are low and they're doing a lot of hand waving about, they're not actually sampling the infant's upper and lower tract. They're saying because they have these antibodies to this virus and this virus we know from animal experiments. So it's interesting, but I think a lot of hand waving. I would love to do that experiment.
So they have a lot of cell culture experiments which show the ability to get in by FC receptors, right? But that was also the case for Zika, and you could never show in people that the ADE actually happened. So they say basically there's very little evidence that this happens in patients at all.
Yeah, so. They also make a couple comments about what's been seen with people getting convalescent serum.
Yeah.
Because in theory, convalescent serum could potentially also give you this enhancement. And they point out that it hasn't really been seen there.
Yeah.
So I think that, we could look very carefully at some of the data as far as convalescent serum to try to address some of this, but I don't think that we've seen any convincing slam dunk evidence here.
With the convalescent serum, though, have they been giving it to the right individuals?
Well, they're in hospital people so far, right?
Okay,
that's the problem.
They're still productively infected.
Well, their disease is not a viral disease anymore, right? So the serum.
Right, so that's why I continue to wonder about. I think they're doing safety trials, but the efficacy part of it isn't entirely clear because the idea with convalescent serum is you're taking antibodies from an individual who survived the infection and giving them to someone who has the infection, hoping that will keep that person alive long enough so they can generate their own immune response and then recover from the infection. But the problem is that for a lot of drugs that you're testing and things like that, you're doing it in very, very ill people.
That's right.
In the case of the SARS CoV-2, those individuals have been in the hospital for a while and they might not actually be virus positive anymore and productively infected. So it's unclear how convalescent serum will be working.
I think that's the problem of giving drugs or monoclonals or convalescent to very, very sick hospitalized patients because it's too late. If you give it earlier, then you may see the effect of antibodies making it better or worse. Daniel said if in a study given to healthcare or people just after their PCR positive test, then it helps if you give it very early. But I agree. Certainly in the convalescent sera tests, we haven't seen a benefit, but there hasn't been any worse disease so far, which is good. But
that's good.
Yeah. So I think obviously it's worth pursuing, and these vaccine companies, really would not want antibody-dependent enhancement to be even a small percent, because when you amplify it by how many people we have to vaccinate, you're going to see that.
And that's what happened with the dengue, that it was such a bad outcome.
Yes. But correct me if I'm wrong, Vincent, that was pretty well established that this happened with dengue before they.
It was clear in dengue, but then they released the Dengvaxia, right? And the trials had looked fine, but then in all the people that got it, subsequently they got enhanced disease with one of the serotypes, serotype 2. Yeah. Clearly,
well, that comes back to who is in your clinical trial. If it's adults,
right?
And in the case of dengue, if you had the natural infection before, you were okay. It was the children who had not been infected before.
Well, they did
any of the serotypes that got the vaccine first.
Yeah.
Then when they were actually infected, they had worse disease. Is that am I correct on that?
They did trial the vaccine in younger kids, and that's whom the vaccine type 2 induced more serious disease because they hadn't looked long enough in the trial because the vaccine was their first infection essentially with that type 2, and then when they got a different serotype within a year or two, then they got serious disease. That was
the problem.
They hadn't looked long enough.
Because it's seasonal, so it took a year.
It's seasonal, and the serotypes vary from season to season as well. So they hadn't waited long enough, and that was the problem. But so
will that be a problem with SARS CoV-2 if it's seasonal?
I don't see any evidence, as Steph said, so far. I don't think so. I think, as Steph said, it's a possibility. That's why people are worried, but I don't see any reason to worry about it.
And I don't know that there's evidence that there's going to be sort of type-specific immunity, that this virus is going to mutate to the point of leading to different types. I'm avoiding using the S word because I know Vincent doesn't like it. So
sometimes I even don't like the M word, depending on how it's used. So far I don't. All right, so the last thing very quickly is very interesting, seroprevalence. And they... So first of all, two things. Average age of your first infection with these common cold viruses, 4.8 years, although that varies according to the study. Some of them are older and so forth. But they have a wonderful graph, figure 6, age seroprevalent curves of these endemic coronaviruses, all 4. They each have a graph, and they're different studies done. And you can see that depending on where you look, there you go. Stephanie's showing it. Depending on where you look, like there's one study in the U.S. in 1970, very low seroprevalence for 229E, but then what is that? One other study, Xiao in the U.S., by 20 years of age, almost everybody is seropositive. So it varies according to the virus and according to where you are. Some are really high.
I don't know that they looked at this, but there's a, and Steph and Brianne will probably realize this quite quickly. There's a huge confounding factor to these different time periods, right? So in the 70s versus now, there's significantly more women in the workforce, and significantly more children going to daycare earlier on. Sure. And so the likelihood that you're going to be exposed is much earlier. So I would look at like the upper right one, where in 2013, very early on, all the kids are positive. Well, that's probably because there's way more children going to daycare now than in 1970.
And the other thing that's important here is that these curves are rising continuously throughout age. And they say that is because you're getting infected over and over. The infections are continuing. So, and that's we did a paper last week on TWIV where they looked at this Amsterdam cohort. Every 6 months, you got infected. Even though you made antibodies, they waned and you got infected again over and over. So probably that's the general. There are going to be exceptions, but.
So I guess what we are taking from this, though, is it's not picking up the granularity of the waning, correct? I mean, really what we're just looking at is a consistent increase. So that's interesting.
It's a 1 point. That's the thing.
But
this is a one.
Seroprevalence is measuring within a population, right?
Yes,
so
it's one time point.
If you become positive, you're positive, you don't go negative again. They just start adding on to it. So over time,
and in the
more individuals are becoming positive.
Right.
Yeah, the Amsterdam was. Sorry, go ahead.
I was going to say the other thing we should mention is that these also, you in some cases use different assays.
Yes, of course.
And so there also are some differences in terms of which assay was used when and how that might relate to what's going on with the data.
Yes. And the Amsterdam study that we talked about, they had samples for many, many years from the same people, right? Because this was the HIV cohort where they would get sampled every few months for years.
Yeah.
And so they had all those frozen and they could just go back and say, so with time, you can see these rises and falls. It's really quite impressive. All right, I need to go. I'm sorry, but I think we did a good job with those two. We learned something.
I think we covered a lot of ground.
We did.
That was great.
That was fun. So maybe we'll think about, a guest, potentially Donna Farber. And I would be neat to have my boss, Sally Permar, on to talk about kids. They just published a viral titer study. And we're going to we have some interesting rhesus infant work that's going to be happening soon. So
okay. So we can, after we're done, we'll pick a date and see if we can get a guest. All right, so that is Immune 35, microbe.tv/immune. You'll find the show notes and when we have letters. We have a bunch of letters. We'll get to them at some point. If you have questions or comments, immune@microbe.tv. And if you want to support us financially, microbe.tv/contribute. Cindy Leifer's at Cornell University on Twitter, Cindy Leifer. Thanks, Cindy.
Thank you.
Steph Langle's at Duke University, Stephanie Langle on Twitter. Thanks, Steph. Welcome back.
Yeah, thanks. Good to be back.
And congratulations on the child.
Thank you. The child. And also Nels Elde, from what I heard from TWIVO, recently had a baby too. Yeah.
That's right. Yeah, he and his wife had a baby, and pretty much the same pandemic babies. Yeah, cool. And Brianne Barker is at Drew University, BioProf Barker on Twitter. Thanks, Brianne.
Thanks. It was great to be here.
So now you are a permanent member of Immune, is that right?
Yay. Yes.
Hope so.
So you want me to leave you three to just go forward and I'll just step back?
We like having you around.
Oh, we like your color commentary.
All right.
Yeah, you should stick around.
Yeah, you need me for the tech, right?
Yes, the tech. Nice. Yeah. And we need a little, we have to have a little grumpy to balance us out.
Yeah, you could figure out. I could teach you how to record, but I'm happy to stick around.
Well,
I like learning.
I mean, if you find yourself, it's too much, you can let us know.
I just like learning immunology. So, it's good to be a student.
It's really fun.
Yep. And I am Vincent Racaniello. You can find me at virology.blog and on Twitter, ProfVRR. Music on Immune is by Steve Neal. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.